Oxford scientists cut brain atrophy by 53% with 3 vitamins. Stop normal aging after 50. In 2014, researchers at the University of Oregon published a study that quietly changed how I think about cognitive decline in my patients. They measured blood levels of 30 different nutrients in a group of older adults, then performed detailed brain imaging and cognitive testing on the same individuals. What they found was striking. The nutrient levels in the blood, not age, not genetics, not education, were the strongest predictors of both brain volume and cognitive performance. People with higher levels of specific vitamins and fatty acids had larger, more metabolically active brains. People with lower levels had measurably more brain shrinkage and performed worse on memory and processing speed tests. Not older brains, not genetically inferior brains, nutritionally depleted brains. Now here's the part that most people over 50 have never been told. The aging brain does not just need the same nutrients the young brain needs. It needs them in higher amounts. It absorbs several of them less efficiently, and it becomes selectively vulnerable to deficiencies that younger brains can compensate for. A nutritional gap that a 30-year-old brain navigates without consequence can, in a 60-year-old brain, produce measurable cognitive effects within months. Brain fog, word-finding difficulty, slower processing, reduced concentration, declining memory. These symptoms are so routinely attributed to normal aging that the nutritional causes are almost never investigated, and yet the research is clear, consistent, and actionable. Today I am going to show you exactly which nutrients the aging brain depends on most, why deficiencies become more common and more consequential after 50, and what specific steps the evidence supports for protecting and potentially restoring cognitive function. We will dive into all of that in today's episode. I am Dr. Alexander Wellrich. The brain is the organ most people are most frightened of losing, and with good reason. The loss of cognitive function is not just a medical event, it is a loss of identity, of independence, of the self. And the fear of Alzheimer's disease and dementia looms over the second half of life for millions of Americans in ways that their doctors rarely address with anything resembling a proactive plan. What I want to give you today is precisely that, a proactive plan grounded in the science of brain nutrition. Not a guarantee, not a cure, but a set of evidence-based interventions that address modifiable nutritional drivers of cognitive decline, drivers that most people and most physicians are simply not paying attention to. We have covered the brain across multiple episodes in this series. We talked about how mitochondria power neurons, how gut bacteria produce 90% of the brain's serotonin, how chronic inflammation drives neuroinflammation, how sleep activates the glymphatic system that clears Alzheimer's-associated proteins, how NAD+ fuels neuronal DNA repair, how sex hormones protect neuronal architecture. Today we add the final layer, the specific nutrients the brain requires and what happens when they run short after 50. If this series has been valuable to you, please subscribe and leave a comment. And now, let us talk about feeding the most complex and irreplaceable organ you have. Before I walk you through the specific nutrients, I want you to understand why the brain becomes more nutritionally vulnerable after 50. Because this is not obvious, and understanding it makes everything else more urgent. The first reason is reduced absorption. Several nutrients critical to brain function, B12 in particular, require a protein called intrinsic factor produced by the stomach lining for absorption. After 50, gastric acid production declines, intrinsic factor production decreases, and B12 absorption from food drops significantly. A person eating the same diet at 60 that they ate at 35 may be absorbing dramatically less B12. The same pattern applies to varying degrees to magnesium, zinc, and several fat-soluble vitamins. The diet has not changed. The gut has. The second reason is increased demand. The aging brain faces mounting oxidative stress from mitochondrial inefficiency, chronic low-grade inflammation, and accumulated environmental exposures. Neutralizing that oxidative stress consumes antioxidant nutrients faster than a younger brain requires. The brain's antioxidant defense systems, which depend heavily on glutathione precursors, vitamin E and vitamin C, are working harder and depleting faster. The third reason is the blood-brain barrier becomes less selective with age. The tight junctions that control what enters the brain from the bloodstream become more permeable, allowing inflammatory molecules and toxins greater access to neural tissue, while paradoxically becoming less efficient at transporting some beneficial nutrients into the brain. This means that the brain's nutrient environment can decline even when blood levels appear adequate. And the fourth reason is that cognitive decline itself is a lagging indicator. Nutritional deficiencies affect brain function gradually, over months and years, before they produce detectable symptoms. By the time cognitive changes are noticeable, the deficiency may have been present and doing damage for a significant period. This is exactly why the proactive approach, assessing and optimizing brain nutrition before symptoms appear, is so much more powerful than waiting to respond. Let me walk you through the specific nutrients, starting with the B vitamin family. Because no nutrient group is more central to brain health, and no group is more consistently deficient in adults over 50. Vitamin B12 is the most critical. It is essential for the myelin sheath, the protective coating around nerve fibers that allows electrical signals to travel efficiently between neurons. B12 deficiency causes myelin degradation, slowing neural transmission in ways that produce memory problems, cognitive slowing, difficulty concentrating, and in severe cases, frank dementia. A study published in Neurology found that individuals with low B12 levels had significantly higher rates of brain volume loss over 5 years. The loss was not small. It was the equivalent of years of accelerated aging in brain tissue. Critically, standard blood B12 testing frequently misses functional deficiency. Serum B12 can appear normal, while cellular B12 activity is inadequate. A more sensitive marker, methylmalonic acid, or MMA, rises when B12 is functionally insufficient, even when serum B12 looks acceptable. If you have cognitive symptoms and normal B12, asking your doctor about MMA levels is a clinically reasonable next step. Folate, B9, works in direct partnership with B12. Together with B6, these three B vitamins regulate a metabolic process called the methylation cycle, which controls the conversion of a potentially neurotoxic amino acid called homocysteine into harmless byproducts. Elevated homocysteine is one of the most consistent biomarkers of neurodegeneration risk in the research literature. A landmark Oxford University study, the VITACOG trial, published in the Proceedings of the National Academy of Sciences, found that B vitamin supplementation in older adults with mild cognitive impairment and elevated homocysteine reduced brain atrophy by 53% over 2 years compared to placebo. Not a modest effect. More than half the brain loss prevented by addressing 3 B vitamin levels. Food sources of B12 include animal products exclusively: meat, fish, eggs, dairy. Vegans and strict vegetarians are at particularly high risk for deficiency and should supplement without question. Methylcobalamin is the form with the best evidence for neurological applications. Folate is found in leafy greens, legumes, and fortified grains. B6 is in poultry, fish, potatoes, and bananas. A few years ago, I worked with a patient I will call Thomas. He was 63, a retired architect from Boston, Massachusetts. Precise man, proud of his mental sharpness. He came in describing a gradual erosion of his working memory, difficulty holding multiple pieces of information in mind simultaneously, struggling to recall names in conversations he had the day before, and what he described as a slowing in his ability to process complex spatial problems that had once been effortless. His primary care physician had attributed the changes to normal aging. His standard lab work, including a serum B12, had come back within the normal range. When I looked more carefully, his serum B12 was at the very low end of the reference range, 218 picograms per milliliter, technically normal but functionally borderline. His methylmalonic acid was elevated. His homocysteine was at 14 micromoles per liter, a level associated in multiple studies with significantly increased neurodegeneration risk. His folate was insufficient. His omega-3 index, which we will discuss in a moment, was low. Thomas was not experiencing normal aging. He was experiencing the cumulative cognitive effect of multiple correctable nutritional gaps that his standard workup had completely missed. We initiated a targeted B vitamin protocol, optimized his omega-3 intake, and added the additional nutrients I will describe shortly. Over 6 months, Thomas reported a noticeable improvement in working memory and processing speed. Objectively, his homocysteine dropped from 14 to 8. His story illustrates the gap between what standard care looks for and what the brain actually needs. But B vitamins are only the beginning, because the research identifies several other specific nutrients that the aging brain depends on in ways that are just as critical and just as commonly deficient. Let me walk you through them. The brain is approximately 60% fat by dry weight, and the most structurally and functionally important fat in the brain is DHA, docosahexaenoic acid, one of the long-chain omega-3 fatty acids found in fatty fish and marine algae. DHA is incorporated into the phospholipid membranes of neurons, where it maintains membrane fluidity, the property that allows neurotransmitter receptors to function efficiently and synaptic connections to form and strengthen. A brain with insufficient DHA has neurons with stiffer, less functional membranes. Synaptic signaling becomes less efficient. Learning and memory are directly impaired. A study published in Alzheimer's and Dementia, the Journal of the Alzheimer's Association, found that higher omega-3 index levels, measured in red blood cell membranes, were associated with significantly larger hippocampal volume, the hippocampus being the primary brain structure for memory formation. Adults with the highest omega-3 index had hippocampi that were, on average, significantly larger than those with the lowest index, independent of age. Bigger hippocampus, better memory preservation. The target omega-3 index for brain protection is above 8%. Most Americans are below 4. Fatty fish, salmon, sardines, mackerel, herring, consumed 2 to 3 times per week is the dietary foundation. For those who do not eat fish, a quality fish oil or algae-based DHA supplement providing at least 1 to 2 grams of combined EPA and DHA daily is supported by the evidence. Choline is a nutrient that receives far less attention than it deserves. It is the direct precursor to acetylcholine, the neurotransmitter most centrally involved in memory and attention, and the one that degenerates most severely in Alzheimer's disease. Your brain cannot make acetylcholine without adequate choline, and choline cannot be synthesized in sufficient amounts by the body. It must come from the diet. The primary dietary source of choline is egg yolks. A single egg yolk provides approximately 150 milligrams of choline. The adequate intake for adults is 425 to 550 milligrams per day. A study published in the American Journal of Clinical Nutrition found that higher dietary choline intake was associated with better cognitive performance and lower risk of dementia in adults over 60. Yet surveys consistently show that the majority of Americans fall short of adequate choline intake, partly because of decades of advice to limit egg consumption based on concerns about dietary cholesterol that the current evidence does not support. Let me cover 3 more nutrients where the brain-specific evidence is compelling and where deficiency is common in adults over 50. Magnesium L-threonate is a specific form of magnesium that was developed by researchers at MIT specifically for its ability to cross the blood-brain barrier more effectively than other magnesium forms. Magnesium in the brain regulates NMDA receptors, the glutamate receptors that govern synaptic plasticity and the cellular mechanism of learning and memory called long-term potentiation. A study published in Neuron found that magnesium L-threonate increased synaptic density in the hippocampus and significantly improved both short-term and long-term memory in aging animal models. A human clinical trial published in the Journal of Alzheimer's Disease found that magnesium L-threonate improved cognitive measures including executive function and working memory in adults over 50 with cognitive complaints. The typical dose used in studies is 1.5 to 2 grams per day in divided doses. Vitamin D, which we have discussed across multiple episodes for its roles in immune function, thyroid health, and bone density, also has direct neurological relevance. Vitamin D receptors are present throughout the brain, including in the hippocampus and prefrontal cortex. Vitamin D regulates the production of nerve growth factor and supports neuronal survival. A meta-analysis published in Nutrients found that vitamin D deficiency was associated with a significantly increased risk of cognitive impairment and dementia. Given the extraordinary prevalence of vitamin D deficiency in American adults, estimated at over 40%, and the brain's documented sensitivity to deficiency, optimizing vitamin D is a straightforward and highly accessible intervention for cognitive protection. Lion's mane mushroom, Hericium erinaceus, is the functional food with the most compelling emerging evidence for cognitive support. It contains compounds called hericinones and erinacines that stimulate the production of nerve growth factor, a protein essential for the growth, maintenance, and survival of neurons. A randomized controlled trial published in Phytotherapy Research found that adults over 50 with mild cognitive impairment who consumed lion's mane extract for 16 weeks showed significantly improved cognitive function compared to placebo, with the improvement reversing when supplementation was stopped. A more recent trial published in the Journal of Neurological and Neurosurgical Psychiatry found similar results in a larger cohort. Lion's mane is available as a food. It can be cooked and eaten, and as a supplement. Doses used in clinical trials range from 500 to 3,000 milligrams of extract daily. Here is your brain nutrition protocol, grounded in the evidence. 1. Request a comprehensive brain nutrition panel at your next appointment. Ask specifically for serum B12, methylmalonic acid, homocysteine, folate, vitamin D, and an omega-3 index test. These are not exotic tests. They are available at most labs, covered by most insurance when indicated, and they give you a precise, measurable picture of your brain's nutritional status. 2. Eat fatty fish 3 times per week. Salmon, sardines, mackerel, and herring are the highest DHA sources. If fish is not accessible or preferred, a quality fish oil supplement providing at least 1 gram of DHA daily is the evidence-supported alternative. Aim for an omega-3 index above 8%, your target, not a threshold. 3. Eat 2 to 3 whole eggs per day, not egg whites, whole eggs, with the yolk, which contains the choline. This is the most practical, affordable, and complete brain nutrition food available. The concern about dietary cholesterol from eggs has been substantially revised in current nutritional science. 4. If your homocysteine is elevated, above 10 micromoles per liter, discuss B vitamin supplementation with your physician. A combination of methylcobalamin B12, methylfolate, and B6 is the protocol used in the VITACOG trial. Do not self-prescribe high-dose B vitamins without baseline testing and physician oversight. 5. Consider magnesium L-threonate at 1.5 to 2 grams per day in divided doses, particularly if you have cognitive complaints, disrupted sleep, or elevated stress. Discuss with your physician, especially if you have kidney concerns. 6. Optimize your vitamin D to between 50 and 70 nanograms per milliliter through testing and supplementation. This is simultaneously a brain intervention, an immune intervention, a bone intervention, and a thyroid intervention. It may be the highest leverage single supplement available to adults over 50. Let me bring everything together. The cognitive changes that millions of Americans over 50 accept as inevitable aging are frequently, at least in part, the consequence of specific, measurable, correctable nutritional deficiencies. B12, folate, B6, DHA, choline, magnesium, vitamin D. These are not exotic compounds. They are fundamental nutrients that the aging brain requires in adequate supply and that become harder to obtain and more critical in their absence as the decades pass. The brain is not a bystander in your nutrition. It is a target organ, one that responds measurably to what you give it. Thomas's homocysteine came down. His working memory improved. The VITACOG trial cut brain atrophy by more than half. The omega-3 research shows larger hippocampi in people who prioritize DHA. These are not soft outcomes. They are measurable, documented effects of giving the brain what it is designed to run on. The proactive window is now, before significant symptoms, before measurable atrophy. That is when nutrition moves the needle most powerfully. If today's episode gave you something you can use, please like this video and subscribe to the channel. Leave a comment. Tell me which of these nutrients you are going to check first. I read every single one. In our next episode, we are going to talk about something that is affecting nearly every adult over 50 in ways that touch their daily comfort, their joint health, and their physical freedom. Chronic pain. The science of what drives it after 50. Why the standard approach of managing symptoms without addressing root causes leaves most people cycling through medications that provide temporary relief without resolution, and what the research says about addressing the underlying drivers of pain rather than just its expression. Until then, take care of yourself from the inside out.